With that, I will introduce our first speaker. The keynote speaker today is Carolyn Hogg. We are super grateful. What? Have I done something wrong? You probably not yet. Maybe I can read. I'm going to read your little bio. I'm very confused, as you can see. And just so Carolyn, we are super grateful she's agreed to come. She agreed to come to the first iteration of this conference and the second one, and now also the third one. So I'm very pleased to have gotten her all across the world. I heard it was a 34-hour flight, so you know, she's going to be up for anything, right? Going for a long run this afternoon after your talk. Yeah, in the meantime. Carolyn, we are super honored to have her opening this conference. She's been working on conservation of threatened species for more than 25 years, both in Australia and overseas. I understand that she started off working in Wales, but now works on all things that are small and brown and hoppy. This includes things like Tasmanian devils, which she's very well known for working on, but also koalas, bilbies, woylies, and more colorful things like orange-bellied parrots. She works closely with both academic and conservation practitioners to create a conservation legacy for Australia that changes the way we integrate science, management, and policy. So without anything else, I am very excited to introduce Carolyn and looking forward to hearing your talk. Thank you.
Thanks, Beth. Okay, hopefully I get this right. So yeah, I think you guys pretty much summarised what my talk's going to be about in your opening address. So that was quite great. There's a little bit of RADseq, a little bit of whole genomes. So today I'm here to talk about conservation genomics, and one of the requests I had was to talk about some of the innovations we've been doing, applications, and where to next. And I didn't really kind of cover innovations. I think all I'm going to talk about today are challenges. We've had some significant challenges in some of the work that we've been doing. Before I start today, however, I'd like to take a moment to acknowledge the first traditional owners of Australian lands in which I work. Aboriginal Australians have a longest living culture in the world at 60,000 years, and I pay recognition and respect to their elders past and present. So I love this photo. For anyone who's ever seen some of my talks, I always use this image. It was taken by Bill Anders in December 1968. And in the 54 years since this photo was taken of our first image of Earth, we've invented Wi-Fi, genome sequencing, artificial intelligence, machine learning, and we've also managed to kill 530 species on the planet. So 530 of the 902 species that are now listed as extinct by the IUCN have occurred in the last 54 years. And I don't think any of us in this room don't recognise the current biodiversity and extinction crisis that we find ourselves in. In fact, finally, the post-2020 framework will be signed off next week in Montreal, in 2 years later after COVID. But what we do know about biodiversity is it's not just important for those of us who like nature. Biodiversity is essential to our own survival on the planet. It's important for our food security, it's important for our health, for being able to develop new medicines. It's also part of the solutions for climate change. I think if I go to one more meeting where people tell me they want to invent a way to scrub carbon from the atmosphere, it's called a tree. Just plant trees. It does a very good job. But it also has both cultural and economic value. So that's part of the reasons why it's important to make sure we maintain biodiversity in the longer term. And one of the ways many of us in this room have actually started working towards this, of course, is the large number of sequencing initiatives that are now occurring about sequencing life on Earth. And Beth said it best in the opening address when she said, well, it's not just about one genome anymore. It's about those genomes at the population level. And although we're getting increases in genomic resources, it's fantastic. We're seeing a very sharp increase as time goes on. There is still an unequal increase in genomic resources. So for threatened species, that's about 15,500 species that have been assessed to date that are listed as threatened by the IUCN. Less than 3% of them still have reference genomes. And today I'll show you some of the stuff that we've been able to do with threatened species when we get a reference genome. But it's something that we need to consider moving forward, is how do we take the technology that we're all talking about today and push it to its absolute limit? Because these are the species that actually require the most assistance on the planet. And this project really started for us when I started investigating threatened species genomes in Australia. So if you look at our 200 national vertebrate recovery plans, About 85% of them require a genetic action. So they say we need a genetic action to help assist with recovery of those species. And at the time when we did the analysis, less than 15% of them were in fact generating or using any form of genetic data. And so that's where we set up the Threatened Species Initiative, which I'm not going to talk about much today, but it really lends into what we're trying to do in Australia. And it's not only about generating genomic resources for Australia's threatened species, but it's also about generating associated population data. And Klaus, I hate to tell you this, but we're going to be stuck in RADseq for a very long time in Australia because sequencing is cheap in the United States and Europe and nowhere else in the world. So unfortunately for us, whole genome resequencing is a lovely pipe dream, which we will eventually get to, but for many of us, the sequencing costs are just too high, so we will stay with RADseq for a period of time. But more importantly, what we're trying to do is develop what we call the Applied Conservation Genomics Portal. And this is really an online portal where conservation managers and end users who know they want genetic data can come to, but it'll help understand, you know, how they're collecting samples, who should they be going to, contact lists, and we've also got the analysis portal where they can bring their reduced representation data at the moment, and it helps run it through a standardised bioinformatic pipeline to help end up with a more standardised conservation report. And the reason for that is the feedback we've had from the end users is that the more they see the data presented the same way, the easier it is for them to start to understand what they're looking at. And so really it's an end-to-end systematic framework from where you collect the samples up in step one, running through sequencing, through standardised reporting, interpreting that information and then actually applying it in real time to the management actions. So currently, this is a bit out of date now, we just went through phase 3. We're doing about 87 different species. You can see we cover pretty much the gambit of threat status under the IUCN. We do, in fact, have 3 extinct in the wild species. And most of the species, taxon groups are in plants, and that's because most of our threatened species in Australia are, in fact, plants, and then a variety of other taxa as well. And it really is a national collaboration. There's about 140 collaborators now, but only a third of them are, in fact, academics. The rest of them are conservation end users. So these are people that work for government agencies or NGOs such as the World Wildlife Fund or the Australian Wildlife Conservancy. And we've tried to spread the number of species across the country as well that we're providing resources for. But it's not just about generating genomes for conservation. We've actually got another project in Australia at the moment, which is called CIPS, which is the Centre for Excellence and Innovations in Peptide and Protein Science. And everyone kind of starts to scratch their head and goes, why is a conservation biologist working with a bunch of chemists? And the main aim of this project is that by 2050, it's estimated that 10 million people a year will die from antimicrobial resistance. And we haven't invented a new antibiotic in the world since the 1950s. And so what this project is about, it aims to decode, to discover, and how to develop new peptides and drugs based on sequence from native species. And then they call it adding on the extra bits that make the peptides nicer, and you can use them. So we've been investigating quite heavily antimicrobial peptides, of course, from marsupials. Marsupials, when they are born, are heavily underdeveloped. And the reason I raise CIPS today, it'll make a little bit more sense later on when I talk about it, but it really is a combination of working with people who are trying to develop new drugs for animal and human health, and how we can work together and leverage off the information we generate for this project for our threatened species. So this is our research group, the Australasian Wildlife Genomics Group. I co-lead it with Kathy Belov, who is currently in Australia. And we've worked on some of the largest genome projects that have ever come out of Australia in relation to Australian wildlife. And so the bottom row are just some of the animals that we're working on at the moment. And how does this tie into CIPS? So what we do with CIPS is we collect samples, we sequence, we assemble, we annotate, and then we go and do peptide discovery. We synthesise the peptides that we discover, and then we go on to test them. But the interesting thing about this is if you're quite strategic in the species that you go and choose for the Centre of Excellence, you can actually select a whole suite of individuals that are critically endangered, vulnerable, or even extinct in the wild. And so we can use the funding for the Centre of Excellence to generate the reference genomes, and then we can use the funding that we have from the Threatened Species Initiative more towards the population genomics and applications. So it's really about marrying together two different fields to leverage what we can for the threatened species. So one of the key things everyone talks about when you talk about reference genomes is sample collection and quality. I like to call it my challenges and collection innovations. So everybody knows if you flash freeze a sample, you get a really high quality reference genome. However, when you work with threatened species, euthanizing and flash freezing samples is not the easiest thing in the world. And I'd like to introduce you to this little critter. This is the Croombat tinker frog. Croombat tinker frogs are only found in one part of the Great Dividing Range just north of Brisbane. They're a high altitude frog. They live in the creeks and streams. Unique to frogs, they don't actually call, so they're very cryptic and they're hard to find. Now the problem with the Croombat tinker frog is there's only 3 adults in captivity, so I won't tell you the reaction I got when I asked them if I could bump one off in order to make a reference genome. But they did tell me, it's okay, we've got some tadpoles that aren't going through metamorphosis. We can knock one of those off for you. So here's the 2 centimetre tadpole that they sent me. And I was thinking, it's a gelatinous mass. How am I going to get a reference genome from that? So we did an DNA extraction of a garden variety frog and we discovered the DNA was black. And it's black because of the pigment in the skin. And so I emailed everyone, Darwin Tree of Life, Earth Biogenome, everyone I knew around the world. No one's done a tadpole because why would you not just kill an adult frog, was the response I got. So Andrew Crawford actually out of Columbia said to me, skin it. So I skinned a 2 centimetre tadpole under a dissecting microscope. That's Elspeth and I. Yes, that is during COVID, but fortunately in Australia we had different rules from the rest of you because we closed our border, so we didn't have COVID for a long time. And we in fact managed to get 45 micrograms of DNA from the circulomics extraction, and we now have assembled the frog genome. It's actually 5.8 gigabases, and now we're trying to figure out how we're going to get tissue samples in order to annotate it. Some of the other sample challenges we've had recently is we decided to do 2 extinct in the wild species that are held under quarantine at Taronga Zoo. This is Elspeth in the white shirt and Emma Peel. They went and took all the equipment out of our lab, drove it over to the zoo so we could do the extractions in the zoo because you can't take the samples out of quarantine unless they're immersed in ethanol, which doesn't work for the tissue samples we're trying to do. Interestingly enough, the extinct in the wild skink species had fluorescent yellow DNA. None of us had ever seen it before, and you can sequence fluorescent yellow DNA, get a really high quality genome. I'll show you in a minute. One of the other things is flash freezing. Flash freezing in the United States is really easy. You guys are geographically the same size as Australia. You've got 350 million people in the US, we've got 23 million. Your road network is a little bit different from ours. We have 5 cities, one in Perth, Adelaide, Melbourne, Sydney, Brisbane. And that's pretty much the only places we can get liquid nitrogen. So we have now got more creative with roadkill, and we got creative with roadkill by purchasing one of these. We've got 2 of these now. These are portable -80 freezers. They plug into any 240 volts. They also plug into a 12-volt car battery. Just make sure you don't turn the car off because it runs your battery flat, as someone from the Devil program once discovered. And that's us working in a shipping container in 1 degree Celsius in order to extract the samples and the DNA for sequencing. So yeah, there definitely are challenges, and you just need to be a little bit creative in order to get the samples. Sequencing challenges. We have 1 PacBio hi-fi machine in our country. 1 machine. We all fight for it. There's now 2. It's great. Ship 2 machines in. So this is work Emma Peel did that got published this year, and this comes from Carolyn going, do I really have to scaffold a genome? Can't I just sequence long read? So Emma went and investigated 8 reference genomes that have been published with a variety of long read, with long read and high C, short read and high C, and the good old 10x linked reads, which is not possible anymore because the technology is gone. But what you can see up here is if you look at highly complex repetitive gene families, and we specialise in immunogenetics in our research group, particularly with MHC, so we do a lot of work with wildlife and disease. You can see when you get the long reads, you get really decent contigs, and then you can use the high C reads to scaffold it, and you can actually end up with these reassembled high-quality genes. It makes life much easier. When you have short-read data, sometimes it flips it around with a high C and you get these weird inversions that occur with your high C scaffolding. And then when you go to actually annotate the gene, you only end up with part of the gene, or sometimes it looks like a pseudogene. And it's simply because the contig is inverted. And don't even use linked reads for immune genes would be my suggestion. So this is the extinct and the wild reference genomes for the Christmas Island blue-tailed skink. This is work that has just come out in preprint from Tristan Dodge. He was a Fulbright scholar out with our lab this year. And the Lister's gecko. So this is actually the skink that had the fluorescent DNA. And the red dots you can see, these are all the contigs that we ended up from the PacBio Hi-Fi, and the red dots are the telomeres. We almost ended up with a telomere to telomere sequencing from our fluorescent yellow DNA. And that's because we actually suspect we ran the DNA through DNA repair and 2 lots of bead cleanups before we sequenced. And so now I want to try that with lots of genomes and to see whether or not I can get away from high C scaffolding. And the reason I want to get away from high C scaffolding is not because I don't like it, but because it's very hard to get the tissue samples in order to do high C. For the Lister's gecko, however, we did need to do high C on that, but we needed to actually immerse the sample in ethanol for 2 days, and then we had to do all these washing steps to try and actually get the high C sequencing to work. We did get it to work, and actually Tristan's done an amazing job with getting 2 high-quality reference genomes for species that now only exist in captivity, and we're starting to use these genomes to develop eDNA markers, and we're doing some population genomics on these species at the moment. This is work by one of my PhD students, Holly Nelson. Same again, this is a critically endangered turtle. Population crashed by 98% as a result of a disease. She's just done a PacBio Hi-Fi genome with high C, so she's been able to scaffold it. What you can see here is the typical microchromosomes that you find a lot in reptiles. And one of the questions Holly's doing is seeing whether or not she can use the Hi-Fi data to actually do the mitochondrial genome. And then can we use that mitochondrial genome to be able to generate eDNA markers? So the question we have for some of these species is, can we use one sampling event to generate a reference genome, mitochondrial DNA, as well as a suite of eDNA markers? So this then leads us to the next challenge, annotation and curation of genomes. What we found with Emma's work with GigaScience is that automated genome pipelines for annotation really work poorly for complex gene families like the immune genes. And this is something as a community, collectively, we need to start thinking about how we're going to better annotate genomes, reference genomes moving forward, because as Klaus said in the opening, functional diversity is actually really critical, and I'll be talking a little bit about that later on. And so this is something that we need to rectify, and what we found is that up to 60% of the immune genes, either the orange bits were incorrectly annotated or the red ones were just not annotated at all by the automated pipelines. And we're only getting about 30% of the immune genes that were correctly annotated. So we're having to manually annotate 70% of the genes. And when you're looking at immune genes, that's about 550 genes in every species that we work with. And so this has massive downstream impacts for us if we're not having high quality genome annotations. The other thing we've got going in Australia, don't ever try and load up to NCBI quickly from Australia. It's 32 hours to fly here. It takes even longer for the data to travel across the Pacific. Sometimes the Pacific cable between Australia and the US is a little bit dodgy, and you get lots of dropout rates. So we've actually started curating all our raw data with Bioplatforms Australia, which actually funds most of our biodiversity initiatives in Australia. And we've got support from Amazon Web Services to have our own open dataset program. This is very similar to where VGP have their genomes in the cloud. So what I want to do now for the rest of the talk is actually talk about how we apply all this information. And the first thing, that's pretty much what koalas do. All day, every day, is to sleep in a tree. But not all sequencing is created equal. And the things that you need to consider when you're looking at population genomics is really things around whole genome resequencing is cost versus coverage. And everyone goes, let's use whole genome resequencing. And then most people do 5 to 10x coverage. And I'm here to tell you, why that's a seriously bad idea. It also causes issues with data storage and transfer. And the deeper you sequence, the more data you produce. The more data you produce, you're going to start hitting problems with compute. The other thing you can also do is target capture, and this is what we tend to do a lot. We do a lot of target capture rather than whole genome sequencing, and this is particularly because of the specific questions we've got around some of the species we work on. But it's also thinking about, if I'm going to capture targets, what am I going to use my data for in the future? It's not just about answering and sequencing for the questions you have now, but also thinking a little bit, like trying to future-proof your data set. But really what it comes down to is, what is your biological question? So, wholesale, whole genome sequencing for all populations is not necessarily the way to go. And the reason for that is, meet my favourite critter. This is a Tasmanian devil. So Tasmanian devils are the world's largest marsupial carnivore. They won this salubrious title on the 7th of September 1936 when we managed to kill off the last thylacine in the Hobart Zoo. And they're not very big, actually, compared to carnivores you have in the rest of the world. They're only about 7 to 10 kilos of male. They're kind of like a size of about a cocker spaniel. They breed from February to March every year. They mate for about 8 days, and they're only pregnant for about 18 days. And they do, in fact, give birth to 30 embryos at once, but they only have 4 teats, and so you only end up with 4 underdeveloped little beans. It's 20-cent piece, is about that big in Australia. And they live in their mother's pouch for about a period of 6 to 8 months, and then they go into the den, and then the juveniles start dispersing in December or February. And they can live up for a period of up to 5 years. So Tasmanian devils went extinct on the Australian mainland about 3,000 years ago. So they're only found on the island of Tasmania. That's a little bit most people forget about. It's at the bottom of Australia. And they've been through a series of unexplained population crashes over the last 20,000 years. So these animals have exceedingly low genetic diversity. And I work with lots and lots of threatened species, and people come to me and say, oh, my species is in dire straits. And I look at the data and I think, it's not a devil. It's fine. It's got more diversity. It's my new benchmark for how bad it can get. Tasmanian devils, however, are now listed as endangered, and the reason is because of this. So this is devil facial tumour disease. It's an infectious transmissible cancer, and they bite it and transmit it from one individual to the next. It first arose in 1996, was where we first saw it, up here at Wakalina, Mount William National Park, and the disease slowly spread south and started moving westwards. And when the disease first arose, they knew nothing about it, and it was killing off populations. We've now seen a decline of up to 80% across the entire species range. We started an insurance population, bringing devils in from 50 kilometres within the disease front, predominantly from the northwest region, in between 2005 and 2008. We got a reference genome for the species, as well as for the cancer itself, produced by Liz Murchison, just down the road at Cambridge University in 2012. And then Liz actually upgraded that really poor quality. The first genome, you should ever go look at the first genome of devils. It's absolutely atrocious by today's standards, but you'll be amazed what we could do with it. But now we've got this fabulous new oxid nanopore genome with high C scaffolding, which is making life definitely much easier. And devils now exist in small fragmented populations. Some populations are starting to suffer from inbreeding depression, and as you can imagine, a carnivore in the landscape, it plays a very, very important ecological role. If we lose devils in the landscape, we know how that story is going to end, because there are feral cats in Tasmania, and in Australia we've lost 39 mammal species since 1788 when we just first brought cats to the Australian mainland. They've basically eaten their way through our wildlife. So up here in Woolnorth in the northwest and the southwest are the only 2 areas at the moment where we don't see the disease. And one of the questions that we've been working on is, should we be supplementing populations to help support them? So the key biological questions we had for our genetic data is really how representative of the wild is the insurance population, and what does wild genetic diversity actually look like? And is it representative at the neutral diversity, at functional diversity? And some people have said there might be some loci that are associated with DFTD resilience. and whether or not the releases that we're doing to these wild sites are actually alleviating genetic pressures within these populations, and whether or not the insurance population animals are naive to DFTD and the natural evolutionary processes that have been going on for the wild for 20 years, and should we actually be releasing them. So this was a massive project. I can't even remember how big Tasmania is. My brain's gone to mush. It's 65,000 square kilometres, I think. So we collected samples from a variety of field monitoring sites. We also took samples from the insurance population. They have the roadkill app. If anyone's ever been to Tasmania, it's the roadkill capital of the world. There is more dead wildlife on the roads in Tasmania than anywhere else. But people actually stop and will take an ear off a devil and put it in a Ziploc bag and post it to us. It's a little bit gruesome, but actually sometimes it does work. And also we flew in here to the southwest back in 2017 to sample animals as well. So you can see we tried to get a representative range-wide example of the population. And this is a little bit of work that was done by Belinda Wright when she was a postdoc in our lab. Belinda was able to show that there's quite strong concurrence between whole genome, genetic diversity, and our reduced representation, what we call DART-seq. It's a type of sequencing that we use in Australia, and also showed that the SNPs that we get from the DART-seq data is actually representative across all the different autosomes and the X chromosome in the devils. So subsequently as well, even with our really bad 2012 genome, we have done lots of cloning and different types of sequencing. We've pretty much characterised all the immune genes in devils, particularly MHC1. And MHC1 is critically important for this species because this is the gene family that's associated with the devil's ability to respond to the disease. And interestingly, DFTD actually downregulates its MHC1 receptors, so the devil's immune system can't actually detect the disease. So based on all that information, we were able to develop a target capture, which is basically you just develop some probes that go in and grab out the genes of interest. We looked at over 500 genes, particularly in the immune gene families, for obvious reasons. We're dealing with a species with a highly infectious disease. But also we were interested in some of the reproductive genes because of the signs of inbreeding depression that we were seeing in populations. And because we could, we threw in all the DFTD-associated loci that had been published as well. So one of the things is you're all sitting there going, why don't you just do whole genome resequencing? Seriously, it'd be much easier. This is why. Welcome to MHC. There are 3 MHC genes, MHC1 genes in devils, UA, UB, UC, and they have 98% sequence similarity, which means if you sequence them with short read data, you can't put them together because you don't know, you can't call haplotypes because you don't actually know where they go. So Wan Cheng, who is a postdoc in our lab, developed a PacBio method which actually grabs all 3 genes at once in one contig, and that's how we sequence it. We looked at this with 45x coverage whole genomes, which have been sequenced with Illumina short read data. We still can't call the haplotypes of 45x coverage whole genomes. We have to do this by target capture, otherwise it's never going to work. And Wan developed a bioinformatic pipeline that uses the PacBio data and a custom analysis piece of software to allow us to be able to call the MHC correctly. So when we look at wild diversity, that's the map of all the sites. Looks like a map of Tasmania. This is a principal coordinate analysis plot for those of you who haven't seen them before. It just basically shows us genetic differentiation between populations. And you can see what we do know is that southwest Tasmania, which are the yellow dots down here, is more closely related to the Hobart area. So the devils are obviously coming into this region. There's a massive mountain range right here. So they're coming in through the south coast. And when we look at the functional SNPs, we still see a little bit of differentiation between the east coast and the west coast, but it's not as pronounced as what we see with the neutral data. If we look at the insurance population, it's pretty representative actually for an insurance population. We've worked really hard to bring in orphans from the orphan program from the east coast, so we've represented most of Tasmania. We're literally missing the northeast, which is where the disease started and populations are very low. The northwest and the southwest are the areas that we don't have neutral representation. And we don't have a lot of the functional diversity that's already been lost from the east coast of Tasmania. So historically, we always thought there was only 2 to 3 genetic populations or genetic regions within Tasmania. But now that we look like there's about 6 clusters, and it really separates between east and west and north and south. And you can see the insurance population and Mariah Island, which is an island off, that's Mariah there, off the east coast, where we actually released animals from the captive insurance population back in 2012. We've made more genetically diverse devils than we've ever seen before on Mariah Island by strategically selecting the animals that go out there for breeding. Interestingly enough, when we look at the DFTD associated loci, there's actually, this is a heat map, these are all the different loci. There's not much difference between populations that don't have disease and those that do have disease. And this leads us to believe that actually these loci are not really real. They're not associated with DFTD, they're associated with some other form of selective sweep that's happening in the wild. There are, however, 3 significant genes, and these are the ones from animals that had the tumour and it's regressed. And 2 of the loci are associated with ACVRB2B. Sorry, it's such a mouthful to say. It's a muscle wasting disease. It's associated with muscle wasting in mice. We think that if you're a devil and you've got a certain type of allele with this gene, it actually allows you not to lose muscle mass as rapidly as others, and potentially that's what allows you to live with the disease a little bit longer. And then we just have a gene. We can see it's a gene. We don't know what it is. So I've got an intern whose entire job in January will be blasting against every known annotated gene on the planet to try to figure out if we can work out what that gene actually is and what it does. Interestingly, when we look at MHC diversity, Weinwein's method was able to call haplotypes. We were able to also look at the different allelic frequencies, so the darker the square and the heat map, the more individuals in those populations have that genotype. And she's been actually, for the first time ever, create supertypes for this. So this work is very recent, and now we're actually going to start using this method to investigate the individuals that we see that have an antibody response in their serum, an MHC antibody response. And so we want to see what supertypes they actually have to see whether or not we can start to figure out how some devils are starting to mount an antibody response and others are not. And this is particularly important for the Menzies research team who are developing the vaccine for this species. The other thing is Southwest Tasmania is the only place that has different MHC diversity. We used to think that there was different MHC diversity between the East Coast and the West Coast from some of our original work with cloning, but the new technology has allowed us to show that although there is more diversity than we originally thought, there's not this structural differentiation in MHC that we originally thought in the populations. So what does this all mean? Like, yeah, it's all really kind of cool pictures of genes and genomes, but how does it actually apply in the real world? So this is what we're trying to do. Working with the government, we've mixed devils on an island and made more genetically diverse devils. And the question now is, can we take devils from there and release them to a location called Stony Head? We've actually got 4 different release sites and 4 different control sites in this project. And what we're trying to see is whether or not we can make a more genetically robust hybrid devil. Can we introduce new genetic material? Can we measure it? And is that having a fitness benefit for the species? So this is work that was led by Weinwein and Elspeth in our group. These are the animals from Mariah Island that we released in the dark blue. These are the animals that existed at Stony Head in the hot pink. And these are the first juveniles in the light blue that were born the year after we released. And so the good thing is we've been able to show the Tasmanian government that at the barest minimum, you can use genetics to see how many individuals actually survived the release and went on to breed, because they produced offspring, and we can reconstruct the pedigree. And we've now got up to date in the wild, about 75% of all juveniles in the wild have some form of mixed parentage between the animals that we released and the incumbent population that was originally there. So what it means is if you've only got stony head parents, even to date, it means you have very high levels of relatedness or mean kinship and low levels of standardised heterozygosity. And if you've got hybrid parentage, you have more genetic diversity and you're less related. It's not rocket science. This is all from the neutral data. It's what you would expect with a genetic rescue event. You would expect to see the decrease in relatedness over time. We did the release in 2016, right about here. And you can see the sharp increase in genetic diversity that you'd see when you do release any new animals to a population, but is in fact maintaining over time, and it's maintaining to F3 generation now. So that's been a success from a neutral point of view. Interestingly, however, when we used our target capture and looked at our 500 genes, we have 4 genotypes that confer a lower neutrophil to lymphocyte ratio in the blood work. 4 different genotypes give us a lower fecal egg count in those individuals, and another 4 genotypes give us lower egg tick counts. And what this means is a lower neutrophil to lymphocyte ratio means you have a better cancer response. We've shown that quite distinctly between both captive animals and animals with DFTD. And if you've got lower parasite loads, you just have better overall immunogenetic diversity. And so, although we can't make devils resistant to the disease, by introducing new individuals and actually looking at the functional diversity using the target capture method, we've been able to really investigate how the genetic rescue event is conferring a fitness benefit for the species in the long term, rather than just looking at the neutral data. The best part for me is we didn't make the disease worse. This is the bit that keeps me awake at night. You know, if I'm going to go dump 30 animals into a population overnight, I'm going to make the disease worse. So what you're looking at here is the years pre-release, and then 2017 onwards is post-release, and this is the frequency. And these are the number of adults who are affected or are not affected by the disease. And the black dots is the prevalence of the disease relative to the size of the population. So you can see the population was quite small in 2016 and not so big in early before release. We haven't made disease prevalence be any worse. It's still ticking around 50% of adults end up with the disease at any time, although we've tripled the population in size. So that gives us some hope that we can use translocations as a tool to help alleviate some of the other small population pressures that devil populations are suffering from because of the disease itself. So what does this mean for devils? Well, we've shown that there's definitely fine-scale genetic structuring across the state, which is great because that bodes well for making a long-term supplementation and translocation plan, particularly for the populations around the coastal areas. Those are the ones that seem to be suffering the most from accumulation of inbreeding and then drop-off in genetic diversity. There's actually more genetic diversity and important functional genes, particularly at immune genes, that we previously thought. So the new reference genome and the new technology is really allowing us to delve down into how we can manage the species at a population scale. We've also got more MHC diversity than we previously realised, which is hopefully good if we ever get the vaccine problem cracked. The insurance population really does largely reflect the wild population, which is fantastic news because we've worked really hard to ensure that that insurance population will be useful moving forward. And that there's no evidence to support some of the criticism we've taken for releasing devils from the insurance population to the wild, that there's evolutionary changes naturally happening in the wild. They don't seem to be naive to the animal, to the system changing, and if the devils don't fully have the high allelic frequency that we see in the wild, they still have those alleles present, which means natural selection will be able to occur when we release them to the wild. And really, this is all going to be used to inform the future actions for the wild devil recovery program. Now we're going to talk about another species, what I call whales of trees, koalas. Everyone loves a koala except me. I don't like them. They bite, they claw, and they smell really bad. But anyway, it's one of my sins in life. I have to work on koalas. I'm going to get in trouble for saying that publicly. Koalas exist all the way down the Australian east coast. This map's a bit old now. The habitat doesn't really look like that. We burnt a lot down here, and it's drying out quite rapidly with climate change. Koalas, however, hit a massive problem in 2019 and 2020 because of this. So the Australian megafires took out huge swathes of land. In the state I live in New South Wales, koalas lost 25% of their habitat. So this is that distribution map again. If we zoom in here, this is New South Wales. It's where I live in Sydney. The green that you can see on the map is what we call areas of regional koala significance, or ARCs. You know, good old government, it's always got to be an acronym. And these are areas where we know koalas exist. We know that there's really good habitat for koalas. And the black you can see is everything that burnt. And we produced a reference genome for koalas. Rebecca Johnson and Kathy Bell have led this work that was published in 2018. And we have a series of management questions from the management agencies post-fires, and it's really about what does genetic diversity look like across the range, what was lost during the mega fires, what populations are at risk of extinction, are populations source or sink populations, can we actually start investigating the genetic basis of disease? Koalas get a lot of chlamydia and also CORV, which is koala retrovirus, so they've got these two major disease issues. And can we find associations between, heat tolerance and phenotypes, taste receptors? We know they've got these really specific taste receptors that allow them to detect water content in the leaves, and some other really functional questions and population dynamics. And then really they also want a whole bunch of information that we can take into account for translocations and manage captive breeding. So I guess I got pulled into working on koalas from the success of working with devils and showing what we could do with devils. So I thought, this is easy. Everyone works on koalas. People have been working on koalas for years. Surely there's a lot of population genetic data for koalas. So in 2020, I got pulled in by the Environment Minister in New South Wales and asked what happened to koala populations and what could the government do to help fix it. So before I went to the meeting, I thought I'm going to do a systematic review like any good scientist. Don't want to go to a meeting unprepared. Surely I can pull some information, I can just amaze them and come up with some solutions. So there's 22 papers published between 1996 and 2020. Of these 22 papers, 17 of them used microsatellites to this day. They use 6 microsatellites. I can see everyone starting to shake their heads. Not only did they use 6 microsatellites that were produced by Bronwyn Holden in 1996, but there's also 4 studies that decided to go off the reservation and develop their own microsatellites. So now we have a whole bunch of data which we can't compare to each other, studying the species across their range. 3 studies, however, before 2020 started to use SNPs, but they didn't use the same type of sequencing. 2 of them used DART sequencing, 1 of them used reduced representation sequencing. Again, we've now generated data sets that are not comparable. This is not useful in conservation at all. It is a real headache because conservation managers go, but we have genetics. They don't understand the minutiae and the nuances of having different marker sets which you can't line up. Since 2020, we have had another couple of studies that have used SNPs. They used exons, again, something different. So it proved out to be a bit of a headache. But interestingly, only 5 of the 22 studies investigated koalas across the range. Most of the studies were investigating koalas just within the state that the researchers were living in. And so what I decided to do I have no idea, is get a whole lot of money from a bunch of people with the Australian government and support from the sequencing companies and Amazon Web Services. And I contacted all the researchers across the range and said, we will whole genome sequence your samples free of charge if you give us your samples and allow us to publicly release the data. So we contacted 49 researchers and 48 of them gave us samples, which is absolutely fantastic. And we have samples all the way across the range. You can see the size of the dot confers to the number of samples that we got to that area. And I'm really pleased to say we got some koalas right up in far north Queensland. I was so impressed by how the Australian research community came to the party on this project. So we aimed to get 20 individuals per region from each of the populations. We got sent 802 samples, and we really requested samples that only represented the last 1 to 3 koala generations. We kind of wanted a snapshot in time to build a data set that we could use now in 2022, but something else that we could also move forward to in 2030, 2035, because the government is about to invest $240 million in Australia to protect koalas. And my question to them is, before you go and spend my $240 million of taxpayer dollars, can we make sure we can benchmark what we do? And so what we're going to do with these populations is then be able to say in 2030, 2035, are these populations better or worse than they were previously? Most of the samples we got were ear biopsies, and we did get some whole blood. We did whole genome resequencing, and we decided to go for 30x coverage. The reason we went for 30x coverage was to not only future-proof the data set, but Luke Silver has a poster, electronic poster, which will be presented, I think, tomorrow. And Luke has shown that if we're not doing 20x coverage, we can't actually call variants very well in immune genes with koalas, so we need to at least be 20x. We sequenced 430 individuals. about half the samples, if you're doing 30x coverage, whole genome resequencing, you need some pretty high quality DNA. And we discovered that you have to DNA repair every sample you get in order to stop the pitting issue we were seeing with the DNA degradation. We do have 17 animals from captivity. Why sequence animals from captivity? It's so we can actually work out the mutation rate. Believe it or not, there is no mutation rate calculated from trios for any marsupial anywhere in the world. So we're going to do it for both the devil's data that we've got as well as koalas. And I would love to show you data, but we only finished sequencing last month. So we've got an average of 32x coverage for all the samples. And this kind of like brings us back to where we started. So we publicly released the data on our genome database. We did about 120 samples each time. We just kept releasing it. So both the FASTQ and BAM files are there. So that equates to close to 60 terabytes of data for 430 samples. So be careful what you wish for when you start wishing for whole genome resequencing. Beth's still whinging at me about my Bilby sample she's got on her computer at UC, telling me that it's taking up too much space. I should send you some koalas. You'll be very happy. And then Illumina built this great, I don't know if anyone's tried the Illumina Dragon pipeline. It's really awesome for joint genotyping. It does 800 human genomes. They said it's going to work for 430 koalas. Promise you, Carolyn, you're not going to have any problems. This is after I broke the national supercomputer 2 years ago. So we ran, so it takes 3.5 terabytes of scratch space to run 800 human genomes. We put in 430 koala genomes and we crashed it in 12 hours and 3 minutes. I take my hat off to the Dragon Illumina team. They have been absolutely fantastic. They have been rewriting their software every time we break it. We've broken it with bilbies now and koalas. Within 3 days, they had recoded the software. The issue is the scratch space. So humans only use 3.5 terabytes of scratch. They made the software tap out and max out at 4 terabytes of scratch, and we went over that with koalas. Koala genomes, the koala genome is only 3.1 gigabases, a little bit smaller than the human genome. The critical problem we've got with marsupials is everything is built for humans and 23 chromosomes. Marsupials have between 7 to 12 chromosomes, so they have these very, very long chromosomes. Hence, stop telling me to scaffold assembly chromosomes in marsupials. They're massive. The bilby chromosome 1 is 936,000 megabases long. It's huge. And no software can actually cope with a chromosome that size. So we sequence, we scaffold, and then we break it apart in order to analyse it. It just actually doesn't make sense. So really, the age of genomics is here. And it's here for biodiversity, which is fantastic because for 50 years we've been able to assess diversity in ecosystems and diversity of species, and now finally we can truly represent diversity of genes. There is a wide range of applications, as we all know, for management and policy and different ecology, but there are some challenges and problems we still really need to solve. We need to solve the problem of long-read sequencing with low-quality DNA. That is something that the sequencing companies really need to be able to crack for us. Somebody else, not our group, but we as the global genome community need to come up with more efficient and effective annotation pipelines. For whole genome resequencing, data storage, curation and analysis pipelines for non-model species really do need to be built if we're going to take conservation genomics to the population level to the next stage. And the ability to multi-thread analysis, I can't stress that enough. At the moment, we can't multi-thread analysis when you do these genomes at scale. And other methods, I would like artificial intelligence and machine learning methods that will start to pull together large disparate data sets in real time. So that I'm talking about if we're producing genomes for a population, there's distribution data, there's habitat data, there's climate data, and a mega fire goes through. We want to be able to pull all that data together in a really quick fashion. It took us 18 months to do that at the moment. I had to pay a postdoc 18 months worth of salary to even get the data started for analysis. I love this photo. I took it in Antarctica. It's a wandering albatross with a giant petrel. And I remember saying to one of my colleagues in the Devil program once, like, what kind of difference has it made to you as a manager having access to genetic data? And he said that having access to genetic data for management of a threatened species is the difference between flying blind and flying with a navigation system. And I think that is a real testament to what we can do for populations, particularly of threatened species at a global scale, moving forward with the technology that we have. So I'd just like to take a moment to thank everybody who helps out and contributes to our program. It's massive, but particularly the Australian Research Council who funds us, and Bioplatforms Australia. We would not be able, Kathy and I would not be able to achieve what we do without all the suite of students and postdocs and collaborators who kind of come on this crazy journey with us on a regular basis. And just to finish up today where I started, which is we achieved amazing things in the last 54 years as a species. We really have big brains and we can really do amazing things. So now we need to figure out a way of turning that big brain to not only saving ourselves, but saving the rest of the planet. So thank you very much for having me today.
Thank you, Carolyn. Thanks, everybody. We have... Nice work. Great opening talk. We have about 10 minutes for questions now before we have a quick break. We have questions that are online that Dario is going to be looking for, and then there'll be a microphone runner for people who have questions in the room. If you just raise your hand, then we'll let you go. I'll start just with the devil's population. We've been following, I mean, a lot of us have been following this story for years. Just what is the long-term plan? I mean, what is the outlook as far as you can tell, and how do you plan to use this in insurance population?
The long-term plan, we have no idea. So at the moment, the long-term plan is to hopefully end up with a vaccine. So Andy Flees runs the vaccine trials down at Menzies, and the plan is to do kind of what you did with rabies in Europe, just throw out baits out the window and have devils eat them. So he's trialling a whole different types of baits. We still haven't got a vaccine that works at the moment, but we're getting closer, edging closer and closer to an understanding. It's something to do with MHC1, we really believe. So that's kind of what we're working on at the moment. And the short-term fix is to use translocations as a way of alleviating pressure in those populations that are on the coast where they're really suffering from interbreeding depression, and then just kind of try and minimise roadkill and dog attacks and other threats.
Yeah. You know, I think one of the things that this project highlights that is something that all of us working in conservation and genomics think about a lot is this tension between maintaining population structure, thinking about local adaptation, and then using translocations to stop.
Yeah, I know that's something everyone else thinks about in the rest of the world. In Australia, we're like, nah, just mix it. To be honest, so with the I shouldn't say that. With reptiles and frogs, we are quite cautious. There are definitely evolutionary significant units. We see that a lot with like the frog species that are found at the tops of mountains. But with a lot of our marsupials, local adaptation is not something we are too worried about. They move in the landscape.
Yeah, I mean, I'm on your side with most of the things that exist. I think I would rather see
with reptiles and frogs, we are a little bit more cautious.
But how do you decide where to draw the line there? I mean, why be cautious with that? Is it just because there's more diversity around, and so you can be, or?
Where do I draw the line? If the species is going to go extinct, then mixing it and making outbreeding pressure and go extinct faster is still going to go extinct. Yeah, that's where I draw the line usually. The orange-bellied parrot. It's a little 40-gram parrot. We got down to 2 females in the wild. We had a captive breeding population. They're monomorphic at all their immune genes. So actually, we're starting to talk about CRISPRing and OBP. I just think it sounds cool as a title for a grant. But we want to use the CRISPR technology that they've developed for chickens to see whether or not we can find genes from historical specimens that we can gene edit in new diversity. If we can't do that, the only other option is to hybridise them with a different species, because that's all we can do. Because what we've shown with OBPs is as soon as we withdraw management actions, the species will go extinct. So it's pretty dire straits.
Yeah, so hopeful for future. There's a question back here. I don't know where.
Yeah, I have the microphone already. So Carolyn,
That's very bright.
It's Camilla here. So very inspiring as always. I don't even know what to ask because I have many questions, but I will follow up
I'm
on
here for 3 days. You can have them.
Yes, I'll follow up on the Tasmanian devil story. So I was wondering how you are following genetically what is happening with the crosses over time. Have you calculated things like the effective population size versus the census? Because I think when you say that the population has triplicated, that's a census, right, as a count? So have you actually, or are you following this from the genetic perspective to know even the pedigree, how they are?
Yeah, so we reconstruct the pedigrees. About 40% of Stony Head site, we've got a reconstructed pedigree for both parents, and then there's about another 30% of the individuals we've got one parent, and then we use Sequoia, which gives us dummy parents. So we can see siblings, but we don't know who the parents are. Because we use reduced representation data, effective population size doesn't actually really work that well. You end up with these massive variances, so it doesn't really mean much the way the algorithm works. But yeah, so we monitor the Stony Head population twice a year. We go out and trap, so we're using capture, might recapture. And on my list of things to do is to actually do the genetics versus the census size. And we can do that using Mariah Island to ground truth it, because it's an island, so it's a closed system. The problem with Stony Head, it's open, so devils come in and out. But if we can ground truth it with Mariah data, we can then start to use it better for the wild populations.
Very nice, thanks.
Quick, Dario, hop in with someone online.
There is just one question online. It's what did you study about whales?
What did I study about whales?
Yeah, this is the online question.
I studied whale snot, believe it or not.
Okay.
I'm a conservation biologist whose PhD is in reproductive biology. I'm not a geneticist. But I came to genetics 12 years ago when I discovered the value of genetics. And so there's two themes in my career. Is I work on threatened species, and I love to use the latest shiny toy in the room. So I've used satellite tracking in the 90s, foraging ecology, stable isotopes in whiskers for leopard seals. But my PhD was actually collecting whale blow. I was the first person to collect whale snot. They do it really easily these days. People use drones. I used a 5-metre cantilevered pole on the front of a boat, nothing like chasing a whale down. And we used to collect the snot and look for reproductive and stress hormones in the blow. And I theorised you could do genetics, but in those days we didn't have the technology for it. So
Has there been genetics now from whale blow?
Yeah, now they use whale snot for genetics, microbiome, hormones. Yeah, it's used all over the world in about 12 different species last time I checked.
Crazy.
I used to be called Dr. Snot. It's such a great title.
Maybe
I think we'll stick with that for the rest of the conference.
Yeah, thanks.
There's two more. There's one in the back, and then...
Yeah, and just following up on Beth's question, I think this is something we can definitely talk about during the conference, about the population structure and mixing versus not mixing, and when to draw the line. So regarding your findings on the structure, do you think it can be anthropogenic, the structure itself in devils? Do you think it's like, there was some structure, right, in your data? Do you think it's original, or fragmentation could have caused some of it?
I think it's just fragmentation over time. If you look at a satellite map of Tasmania, a third of Tasmania is the World Heritage Area. Down in the southwest corner, and then the rest of it is highly fragmented between forestry and farmland, and I think that's where the structuring is coming from. The thing you've got to realise, like, we've got genetic structure in devils, but if you've ever looked at devil data, it's literally like clones. Like we call it the game of clones sometimes when we're trying to figure out who's who, and that's particularly in the insurance population. We really struggle sometimes to even identify parents in group breeding because they're so related to one another. So I think we're picking up a structure, but it's just natural drift, the separation of populations. I don't think it's anything too critical.
Would you think it could be anthropogenic as well?
Yeah.
That's, I think, the question. Yeah. So there's very low diversity, low structure, and the little structure there is could be anthropogenic as well.
Yeah, probably from all the farmland.
Yeah. That's important.
But it's enough structure to make the populations different that when you mix them, you get a bump in genetic diversity.
Right.
So.
But that's exactly what it is.
Yeah,
when to draw the line, the cause of the structure and how important it is to protect it versus undo it.
Yeah, no. Right. And so what we say to the government all the time is, if you don't want to pick up the animals and move them, just protect the landscape between the two populations. But actually, it's cheaper to pick them up and move them.
Yeah. Thank you.
Here in the front.
Thank you so much. It's a very great talk. My question goes a little bit into the weeds because I'm really curious about this. You mentioned several times in a couple of the projects that doing DNA repair really helped generate high quality data. And I just wonder if you could talk a little bit about what you're doing there and what you found to be effective.
So I had no idea what DNA repair was. And the sequencing company are like, can you do DNA repair? And I'm like, what's that? And so actually the way it was described to me by the team at Illumina is that imagine you're driving down a road, it's got lots of potholes in it, and every time you hit a hole, the car jumps. So if your DNA is degraded at any extent, like when the sequencer runs over the DNA, it's jumping, and that's where you end up with these really high error rates in the sequencing that you're doing. And so DNA repair is like driving down the road and spack filling in the holes. And so it just smooths out the DNA and it makes it run much better through the sequencer. So we end up with a lower error rate. And the other thing we discovered with that Koala project, which I highly recommend for anyone who's doing population genomics, whole genomes resequencing, is we were going for a set target of coverage. And so rather than doing, 24 individuals on a flow cell, we did 48 individuals per flow cell, and we would sequence 48 individuals, and then we'd look at their coverage, and then we'd adjust the pooling on the second cell to make sure we'd get consistent data. And when we did that, we were just able to crack consistently hitting 30 to 40x coverage, and that was something that the sequencing guys recommended, and it worked out really well.
All right, well, we have used up our time for questions, and now it is time to have a short break before coming back in here for the second session. Thanks, everybody, for your great questions, and thanks again to Carolyn for a really awesome first keynote. Thank you.